The next talk is by Alexander Berry and co-authored with Ann Ripstra, Altered Gene Expression in Response to Predator Stress in a Wolf Spider, Pardosa milvina, Araneae, Lycosidae, and they're both at Miami University. So take it away, Alexander.
Okay. Screen should be shared. Everyone see everything okay? Hear me okay?
Yeah.
Yeah, you can go ahead and maximize your PowerPoint.
Cool. I'm trying to do that. There we go.
Yeah, you're good to go now.
Awesome. Okay, so yeah, we're talking about altered gene expression in Pardosa milvina in response to predator stress. So I don't think I have to tell a room full of biologists, but phenotypes are rarely as simple as one gene acting upon an individual and determining the phenotype. You often have suites of genes, and of course you have the environment, which acts in concert in many cases with the genes of an individual to determine that phenotype. You have a lot of, again, non-genetic factors that influence the phenotypes. And one of these things could be gene expression. So how many times the gene gets expressed, looking at things like the number of RNA copies, and eventually how many proteins get produced by that individual gene. So there's been some increasing effort to look at gene expression across generations. So not just how does the environment impact the individual who actually experiences that environment, but how does the environment impact their offspring and then potentially grand offspring and further. So I'll be talking about transgenerational effects. And for this talk specifically, I'm going to be transgenerational effects. I'm going to specifically refer to any impact of the parent's environment on offspring or grand offspring, so on and so forth. So you can see in this little cartoon here, we have the normal looking parental dog who has a normal black nose, and then he experiences some kind of environmental stressor, and his offspring have phenotypic change to a white nose. Particularly for today's study, the transgenerational effects you're going to be looking at, obviously, gene expression. So one of those environmental stressors that gets a lot of attention is, of course, predator stress. So we know that predators can have a wide variety of effects on individuals beyond just killing and consuming them. You see a lot of behavioral and physiological changes. You can see shifts in life history, maybe individuals growing faster or not being as large. And many of these things could be mediated through possible gene expression changes. If the predators are altering gene expression in either the adults or in their offspring, that could account for some of the behavioral and physiological changes that are seen. So to introduce you to the star of today's show, it is this wolf spider here, Pardosa milvina. This is a small generalist wolf spider. They live in agricultural fields, pretty closely associated with human habitation. They show a very stereotyped, very well-studied response to predator cues from the larger wolf spider, Tigrosa helio. You see things like reduced movement, reduced foraging, and increased vigilance behaviors. And there are some documented transgenerational effects as well. So you see perhaps reduced movement or increased survival when exposed to a Tigrosa helio in Pardosa milvina whose mothers had predator experience, but who had never experienced predators themselves. So this led us to today's questions. We'll ask you about transgenerational gene expression. So two main questions. If I expose a spider to predators, do we see gene expression changes in that individual? And do we see gene expression changes in that individual's offspring as well? So we're going to be looking at both predator-exposed spiders and their offspring, and those offspring will never have seen predators themselves. So what did we do? We went out into the field, we collected spiders, very young juveniles. We started exposing them at the third instar phase, and we exposed them to predators, predator cues, I should say. And when I say predator cues, these are chemical cues, primarily consisting of things like silk and excreta, other things that the Tigrosa helio just naturally leaves behind it. And we exposed them, the exposure group to these cues twice a week for an hour at a time. And concurrently, we put the control group on just blank filter paper. Again, we collected the diagnostic cues, exposed them for an hour at a time, control on blank filter paper. Once these spiders reached adulthood, we pulled out some of the females used for the genetic to use for genetic analysis, but most of the females we then allowed to mate, and all the matings were done with predator naive males. So the males had never seen predators. The only thing that varied was that the females had, and we allowed them to produce young. And the young of these females were then used in transcriptome analysis as well. Again, the first hypothesis we're testing here is that exposure to predator cues will cause changes in gene expression levels of the actually exposed Pardosa milvina. And the second one is that we will see maternal exposure to predators. So if mom was exposed, we will see gene expression changes. Now, we took 6 predator exposed and 6 unexposed adult females. And again, these were virgin females that we used here. These spiders were not mated. And then we took 6 third instar spiderlings from exposed mothers and 6 from unexposed. We extracted mRNA using a Promega Maxwell simply mRNA kit. And then from those 6 extractions, we constructed 3 libraries per group. So we used the 3 extractions that gave us the best results. The libraries were then sent off in sequence by Novogene, which is a company that does genetic sequencing. And we compared the levels of mRNA expression between the exposed and unexposed adults. And then we compared the third instar spiderlings from exposed and unexposed mothers. So we also did use BLAST software to try to identify the function of a lot of these differentially expressed genes. Of course, a lot of them come back as function unknown, but we were able to identify a good number of them. So to jump into the results here, I think we've already seen at least a couple of volcano plots earlier today, but in case you're not familiar with how they work, along the x-axis, the log two-fold change. So the closer to, in this case, 20 you get, the more mRNA is expressed in this case, the exposed adults compared to the control adults. As you get down to minus 20, there's fewer and fewer mRNA copies in exposed versus control adults. And on the y-axis, you essentially just have your significance level. Just 1.3 is the equivalent of 0.05. So in adults, we saw about 133 genes upregulated, so more mRNA copies in the predator-exposed individuals, and those are shown in red. And we saw 776 downregulated genes, which are shown in green. If we jump to the juveniles here, we see a very similar graph. We see a similar number of upregulated genes, 178. We do see far fewer downregulated genes. In the juveniles, there are only 111 downregulated genes. What was perhaps interesting to me, at least to me, is that out of all those differentially expressed genes, only 13 were differentially expressed in both adults and in juveniles. And some of the functions of these genes, there were a couple of venom genes that were upregulated in both, and there were a couple of things like DNA stability and nuclease binding genes that were differentially expressed in both groups. But again, there were only 13 genes differentially expressed in both adults and their offspring. So if we start to look at what kind of genes, what are the functions of these things, we'll start first with the upregulated genes. In both adults and juveniles, we did see venom genes being upregulated, which makes some sense. If you're surrounded by predators, maybe having more venom or more potent venom is advantageous. We saw metabolic rate or activity genes upregulated in both groups, perhaps not surprising. In the adults, we did see response to chemical genes linked to chemical stimuli were upregulated. This makes a lot of sense because we were exposing these spiders to a chemical stimulus. And in the offspring, we saw drug binding genes Obviously, these were genes analogous to mammalian drug binding genes, although they very well could also be linked to some kind of chemical binding sites. We also saw some other things, stress genes upregulated in the offspring, which makes sense. And then serine protease inhibitors, some cancer genes, and RNA biosynthesis being upregulated. If we look at genes that were downregulated, in both groups, we see DNA repair being downregulated pretty dramatically. In both groups, we do see zinc finger proteins being downregulated. However, I do want to point out that there were many more zinc finger proteins downregulated in the adults. It was the single largest category of downregulated genes in either group, and there were only a fraction of that number downregulated in the offspring. This is interesting, at least to me, in terms of gene expression, because zinc finger proteins do play an important role in regulating gene expression. So these genes could have been impacting many of our other genes we see here. Some other interesting findings in offspring, we see apoptosis and heat shock genes were downregulated, myosin production and clotting in the adults, and RNA polymerase II in offspring. So, and then we look at molecular pathways. The adults, we only see one pathway differentially altered. It was an upregulation in vasopressin regulated water reabsorption. And then in the offspring, we saw a number. Again, we see a transcriptional misregulation in cancer, going back to that, probably that DNA repair stuff. We see an alcoholism pathway. I don't actually think we're producing drunk spiders, but that might be an interesting future study. It probably goes back to that drug binding stuff and maybe linked to some of that chemical binding that we may be seeing in the offspring. So to wrap things up, we saw 909 differentially expressed genes in the adults, 289 in the offspring, and this was not a subset. They were largely completely different genes. And these genes were linked to functions we might expect, things like stress, metabolic rate, chemical sensitivity, and metabolism. I'd like to thank a whole lot of people. First, my committee, especially my advisor, Dr. Ripstra, Miami University for all the support and funding, American Arachnological Society for some funding and allowing me to present here, and a whole slew of undergrads and graduate students who were helpful, and Dr. Kiss and Dr. Moore for helping me with the, particularly with all this, because I'm not a geneticist by training. I'll be happy to take any questions.
Thank you, Alex. And thank you also for staying on time. We do have a couple of minutes for questions. I had one for you, which was, what was the variability amongst exposed individuals like?
Variability amongst exposed individuals, spiders. There was a decent amount of variability. If you look at them, they do all clump into pretty distinct groups. So all the exposed individuals are in one and unexposed. But there was a I don't know exactly how much, but there was a decent amount. There was some, sure. I don't know if how well that answers your question.
No, it does. It does. I mean, I was thinking maybe that there was a few, there might have been some genes that were strongly overexpressed in some individuals and not at all overexpressed in others.
Oh yeah, there was variation. There was some genes that were really overexpressed in maybe one individual and less so in the others. But
sure. Let's see, there's a question. You only used males that were unexposed. Did you see no effects from male exposure? That was from Blaine Hebert.
Okay, so we only used males that were unexposed because for this particular study, we were trying to narrow in on effects of maternal exposure. We have not tested what happens with the male exposures yet. But that might be that is definitely another question to look at, another interesting thing to test. But we were trying to focus in on maternal exposure.
just to let everybody know, we're going to keep everything on time. So the next session will be starting in about 7 minutes in this same room, just so people are aware. And that will give us then the full 30-minute break for after this next session.
Prasad asked, do we know about transgenerational effects in spiders that are ambush predators versus wandering? Wondering if you would care to speculate on how those data sets would compare.
So do we know anything about ambush versus wandering? Off the top of my head, no. I'm hopefully there were people studying that. It's definitely interesting, and how would I think they would compare? I really not entirely sure. I think we might see some similar things. I think, like in this case, I think things like stress and like the metabolic rate of the venom might still be upregulated. Although some other things, like I'd expect things like movement to be less affected, perhaps. We do see fairly strong movement. We found it movement effects in at least the juveniles. We might expect that to be less affected in ambush predators.
Okay. Yeah, that pretty much concludes the time we have for this session. Thank you to all the speakers, and you have just about 6 minutes to either switch around